Good morning, everyone. I hope you're having a great day. It's Friday the 1st. I hope that everyone had a good Halloween if they elected to do anything for that. Today is our last day before our exam on Monday. I've got a couple of emails that folks asked me about extending the window for the exam, and I'm going to do that, but I've got to coordinate, the exact time. Um, And so I'm going to extend the exam window a little bit for Monday, but it's going to start, I think we got it starting at 8:30 in the morning. It's just like our other exams where you've got to use Proctorio, which if I remember correctly, everyone's found their kind of steady state way of working with Proctorio. Um, so we're good there. Now, with that said, you've got your practice exam up. I would do that practice exam. I would also jog through that video that I posted where I walk through the practice exam. Um, can anyone confirm that they saw the link for the walkthrough video? Just put a thumbs up or something in the chat. Okay, so nobody saying anything, so I might have to go back and take a look at that. I thought that I posted that. Um, but I'll make sure. I posted a video where I walk through the practice exam for you. So I'll make sure that's available for everyone. Um, but that's kind of my thoughts on the exam from the jump. We'll talk about it after a little bit though, because what we're going to do today is we're going to talk about enzymes. We're going to introduce enzymes. Um, there is a lecture. I believe that I posted learning objectives. Let me take a minute and I will look at my Canvas page real quick. Page. Oh, okay. Yes. Okay, cool. Yeah. Yeah, so those have been up yeah, those are up. So, um, and then I believe that the walkthrough video for the practice exam, here's a link for the practice exam. Yeah, okay, good. On October 23rd, I posted the link for the practice exam as well as a video where I walk through that practice exam. So I would watch that because I go through some suggestions on, here's the question that's provided in the practice exam. Um, when you are anticipating the actual exam, here's how that question's going to be, or here's a way that you could kind of formulate your own question from this. Um, The material that will be bumped to the next exam that's on our calendar is transition states. And so what we're going to do today is going to be the last of our new material, and that is going to be we're going to talk about enzymes. Now, for chat, sorry, just have to make it. On October 23rd, I posted an announcement. And it says practice exam. I think the subject of the announcement is practice exam. If you go to that, um, you'll see the link for the practice exam as well as the link for the video that has the walkthrough. Absolutely. Absolutely. I'm thank you. I would thank, sorry, thank you for asking because the display that I have on Canvas is different from the display that you guys have. So it's easy for me to walk through, or it's easy for me to see things like, oh, here's the chronology of things that I posted. It might be a little bit different for you. So I appreciate your patience with me in asking these questions. Okay, so enzymes. What does an enzyme do? Now, an enzyme, for starters, um, An enzyme is typically a protein, not always, because there's something known as a ribozyme that is a non-amino acid based enzyme and it has catalytic activity. The I think very common when someone talks about an enzyme, though, um, it's it's pretty typical to be referencing something that's based on amino acids. And now I want to clarify that that is not exclusive, but that's I would say the large percentage of enzymes are amino acid based. So most enzymes are proteins. So not all proteins are enzymes, and not all enzymes are proteins, because some proteins, like hemoglobin, have no enzymatic activity. Now, what an enzyme is it is a entity, it is a something that catalyzes thermodynamically favorable reactions, causing them to proceed at extraordinarily rapid rates. You could also think about an enzyme simply as a biological catalyst, as opposed to an organic or an inorganic catalyst. Enzymes provide cells with the ability to exert kinetic control over thermodynamic potentiality. So they can manipulate the rate at which things go to make something more thermodynamically favorable. Now, living systems use enzymes to accelerate and control rates of vitally important biochemical reactions. So your metabolic reactions, or many of your metabolic reactions, enzymes are the agents of metabolic function. Now, when it comes to enzymes, there are a total of six, or historically, there are six major classes, and I'm putting a check mark by 1 through 6. Now, if you are um, looking to take the MCAT or something like that, you might see, oh, well, there's actually a seventh, and you're right. Now, the seventh item here would be, I'm going to list it down here, 7. They are a class of enzymes that are known as translocases. Now, translocases have really just been added in the past few years. What's significant about the translocases is that they have activities that, or their function and their activity is found in all of, or many translocases have functions that are as oxidoreductases or transferases. Basically, the seventh grouping, these translocases, they have one of these other functions in their kind of purview. Um, so it's not to say that they're irrelevant, but instead they are, I always kind of think about them, or the way that I kind of have thought about them is almost like they're a greatest hits album, in the sense that your enzyme classes 1 through 6 are kind of the foundation. They're very descriptive. Translocases are like they are enzymes that move something across a membrane, but the way that they do that is by exhibiting or having oxidoreductase activity and hydrolase activity, for an example. Now, we're going to be focusing on these first 6. So these first 6, I want you to know this number and connected to this name. So these numbers are specific for these different enzyme classes. Now, when it comes to, oh, not that. When it comes to looking at different enzyme types and how does this apply, there's a couple of ways that you could think about this. One, over the course of this semester, we've talked about a couple of different enzymes. If you think back to when we talked about glycolysis, if we took something like hexokinase, our first enzyme in glycolysis outside of the liver. Hexokinase has an established function. Now, if you were to look at hexokinase in humans or Homo sapiens, or in E. coli, or in yeast, Saccharomyces cerevisiae, what you'd notice is that hexokinase enzyme, it's the enzyme, the protein sequence, the primary protein sequence between all of these, they're pretty highly conserved. They're going to be fairly similar to one another. Okay, so that's one thing that makes them similar. The protein sequences are similar. Now, if we were to look at the enzyme mechanism, and what I mean by that is, take yourself back to organic one or two. I think it was probably a little bit more in organic two. I know in my organic two class, we talked a lot about, and we use this phrase, pushing electrons. Now, I bring that up because if we were to look at the enzyme mechanism for hexokinase in yeast, E. coli, and humans, the electrons that are being pushed and the um, amino acids that are participating in those electrons being pushed, those are going to be virtually identical. Now, that doesn't mean that the primary protein sequence has to be identical, nor the secondary or tertiary structure is absolutely identical, but the movement of electrons to convert glucose plus ATP to glucose 6-phosphate, I'm just going to say G6P, and ADP is going to basically be the same across all of these different organisms, Homo sapiens, E. coli, and Saccharomyces cerevisiae, just as an example of three. Now, the point is, what is this enzyme that has a unique series of movements, of electronic movements. And for that, we give it a enzyme numbering scheme. And that enzyme numbering scheme is going to be a four-character or four-character grouping number. So if we look down at number 2 here, the class 2 represents the transferases. Now, transferases are enzymes that transfer carbon, nitrogen, or phosphorus-containing groups. One phosphorus-containing group, or an example of a phosphorus-containing group, would be phosphate. And phosphofructokinase is an example of a transferase because it transfers a phosphate. Phosphofructokinase, if you remember, transfers phosphate from ATP onto fructose 6-phosphate to make fructose 1,6-bisphosphate. So here's that reaction displayed. ATP, fructose 6-phosphate, making fructose 1,6-bisphosphate, and ADP. Okay, so that's what a transferase does. Now, that 2, that leading number, corresponds to this class. So that leading 2 says that this is a transferase. The next digit, or the next 2 digits, I'm sorry, I'm going to jump back because then we'll be able to see what that is. So, or actually, I'll go back again. It was 2.7.1.11. Okay, so when you look at a table like this or a chart like this, this is the systematic classification of enzymes according to the Enzyme Commission. Now, the Enzyme Commission is the group that ultimately decided, hey, we need a 7th group because these translocases are unique enough that they deserve their own classification, or it's best to classify them with their own separate grouping. Now, if we look at 2, the first leading digit tells us we're talking about transferases, but then we're going to narrow it down a little bit. And we're going to say, okay, 2.7. Well, 2.7 is a transferase that transfers a phosphorus-containing group. So if a protein is transferring a diphosphate, well, that is a phosphate-containing group, so that enzyme is going to get 2.7. Now, to get more specific with them, 2.7.1, that is an enzyme that is a transferase. The 2 is awarded for that. It transfers a phosphate group, 2.7. And then it transfers a phosphate group to a alcohol group as the acceptor. And that's where we get 2.7.1. So then, let's jump back. That establishes our 2.7.1 point. But this last one is for phosphofructokinase. That 11 is a unique identifier for this enzyme. So now, using this information and this 2.7.1.11, add to the chat what you think would be a good scheme or identifier for an enzyme like hexokinase. And I'll present it as a couple of different things. One thing is, would it have a leading 2, knowing what we know about hexokinase's reaction? Then, would it get a 2.7? Then, would it get a 2.7.1? And then last but not least, would it also get 0.11? So I want to present to you those questions and add to the chat what you think a 4-number or 4-character identifier for hexokinase could be. So let me rephrase my question. I'll ask a more discrete question. Hexokinase. We know what that enzyme does. My first question is hexokinase a transferase? Add to the chat yes or no. Perfect. It is. So when we talk about hexokinase and we're talking about the hexokinase reaction, we're going to designate it as 2. Now, if we go back to the previous slide, maybe that will be helpful. I think it will be helpful. When we go back to the previous slide, all transferases get that 2. Then, what do we know about the group that hexokinase is transferring? Well, is hexokinase transferring a carbon group? Yes or no? No. Perfect. Okay. Then let's jump down here. Is it transferring an aldehyde or ketonic residue? No. Then now let's just kind of cut to the chase. Is it transferring a phosphorus-containing group? Perfect. So we're going to start out by identifying hexokinase as 2.7. I always kind of think about this as like a phone number for a specific enzyme or like an IP address or something like that. Okay. Now, when we think about where is that phosphate group being transferred to? Well, it's being transferred to glucose. And so with that glucose, what do we know about glucose's structure? We got a bunch of CH2OH. There's an OH, OH, OH. We got a bunch of hydroxide groups there. Now, my next question is, does glucose have an alcohol group that can accept that phosphorus or that phosphate containing group? Yeah, it's got, we're looking at five of those alcohol groups. So because of that, we're going to say that hexokinase, we'll classify it or we'll give it the kind of phone number of 2.7.1. Now, my last question is, well, we need the specific phone number for hexokinase. And I pose this as an open-ended question, but this last digit, what we do know, and kind of by process of elimination, is, well, phosphofructokinase is a different enzyme than hexokinase. And so it's not going to have the same identifier as hexokinase. So really, we could put 2.7.1.1, point 2, whatever. Anything but 11 at this point, given what we know. Does that make sense? Okay, awesome. So when we think about, I'm going to erase these ones and twos, because I don't want anyone to think like, oh, hexokinase is 2.7.1.1. But instead, what I want you to think about is how could you anticipate or what's the importance of this sort of question? Well, the importance is it gives you a way to classify enzymes. It gives you a way to pose the question of, well, is this enzyme involved in the transfer of something? If the answer is yes, would we also have to remember the six specific subclasses? Okay. Um, no. Thank you for asking that, Milo. You are not expected to know the subclasses. I just want you to know the six major classes. Um, because as you can see, when you look at this, I think that 2.1, okay, that's a couple. 2.2, 3, 4, 5, 6, and we stop at 7. Um, there's a little bit of news for you. There's more than 7. And for all of these, there's more subclasses. So if I were to ask that sort of question, that would be particularly awful of a question. Not only because why, but it would be like, oh my gosh, this is brutal. That's like memorizing the phone book. Okay. So I like to use a transferase as an example because we've seen transferases when we talked about glycolysis. Now, there are a couple of things that are also helpful to think about whenever you're looking at these different classes, these six major classes. The first class are your oxidoreductases. So one corresponds to an oxidoreductase. Now, an oxidoreductase is an enzyme that's involved in an oxidation reduction reaction, or basically any oxidation reduction reaction. Now, an oxidation reduction reaction, I don't know, I would venture to say that most or a large percentage, the largest share of these, of the 7 classes, the largest share of enzymes are probably going to be oxidation reduction reactions. And a hallmark of an oxidation reduction reaction is to look for a molecule like NAD plus or NADH. And if you've ever taken a like cell biology class, you probably learned about the Krebs cycle. Now, in the Krebs cycle, you generate NADH and NAD plus, or you utilize NAD plus and generate NADH. There's another um, electron acceptor, and it's got this similar sort of name. It's all capital letters. NADH is, or NAD and NADH are nicotinamide adenine dinucleotide. Does anyone know of, thinking back to the Krebs cycle or the tricarboxylic acid cycle, does anyone know of any other electron acceptors in the Krebs cycle? I'll give you a hint. The ADH is also there. Perfect. Exactly. FADH2, flavin adenine dinucleotide, is another major electron acceptor. Now, I bring that up because whenever you hear the phrase electron acceptor, I think that it's good to think, oh, okay, something is accepting an electron. Oh, when we talk about accepting and losing electrons, we're talking about oxidation and reduction. When something accepts electrons, it gets reduced. When something loses electrons, it gets oxidized. Now, oxidation reduction reactions are where a huge amount of energy gets harnessed and basically gets captured for the generation of ATP. And I kind of alluded to this when we were talking about glycolysis because I was like, hey, ATP is cool and all, but this NADH, that's going to be super impactful when we talk about the oxidative or the electron transport chain and oxidative phosphorylation, which we're going to talk about that after we talk about lipids. But whenever if you're thinking about like, what should I associate? I would associate NAD, NADH, FAD, FADH2. I would associate those with the number one and the term oxidoreductase. Because if you see, oh, this enzyme requires NADH or NAD plus, well, then that is an enzyme that's going to be an oxidoreductase. So it's going to get one as its class. And then another combination of three other numbers to indicate the subclass of like what's the substrate, what's the electron acceptor, and everything like that. Okay, so one is oxidoreductases. Two are your transferases. Three are your hydrolases. I'm going to clear out all my drawings there. Now, your hydrolases, well, these are enzymes that use water in order to break bonds. So hydrolases. They Urease is an example of that. Urease is an enzyme that uses water to break urea into two molecules of ammonia. Now, your hydrolases, you're going to find those quite often whenever we talk or when you take metabolism and you talk about breaking down a protein and the steps involved in that. Now, lyases are a class of enzymes that what they are going to do is they are going to cleave carbon-carbon, carbon-sulfur, and some carbon-nitrogen bonds. Aldolase is an example of a lyase. So if you remember aldolase, that's our enzyme that was reversible, but it was kind of surprisingly reversible because that took our 6-carbon fructose molecule and split it into two 3-carbon molecules. It split it into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So a lyase, and if you think about aldolase as an example, does anyone remember, given these options, carbon-carbon, carbon-sulfur, or carbon-nitrogen, what bond, which type of those three bonds did aldolase break in glycolysis? And to visualize kind of that F16BP, there's our fructose 1,6 bisphosphate. What type of bond are we breaking with aldolase? Well, with aldolase, what we're doing is we're taking that molecule right here, and we're kind of splitting it down the middle. And we're going to end up with an aldehyde and a ketone, or an aldose and a ketose. So aldolase, absolutely, breaks a carbon-carbon bond. Okay, so lyases, whenever they do that, though, one thing that's important to recognize is what distinguishes them from hydrolases. Water is not required for this. So that's the one of the distinct things that differentiates a hydrolase and a lyase from one another. Water is not required. So H2O not required. Now, isomerases, number 5 is your class for your isomerases. And those I always enjoy because if you know the name isomerase or isomer, you kind of know, okay, well, if I'm talking about an isomer of a molecule, that means there's another isomer of that same molecule. So it's just kind of a rearrangement. And anytime you see that ASE suffix, that's typically an indication that you're talking about an enzyme. An isomerase, well, it's just going to reconfigure or reorganize my molecule in question. We talked about 2 different isomerases. Phosphoglucose isomerase, which rearranged glucose 6-phosphate to fructose 6-phosphate. That was our second step in glycolysis. And then phosphoglycerate mutase, another isomerase that rearranged 3-phosphoglycerate to 2-phosphoglycerate. Our last of our 6 classes that we're going to talk about is the ligases. Now, ligases are valuable because um, these are the types of enzymes. If you've taken a molecular biology class, or when you take a molecular biology class, you'll learn about DNA ligase, and that's a molecule that basically fixes nicks in DNA molecules. Um, and this is also the inspiration for my City League soccer team as a graduate student. We were the ligators, um, or alligators. I can't remember exactly, but the ligases are represented by the number 6. An example of a ligase, or what a ligase does, is it forms a bond between carbon and oxygen, sulfur, or nitrogen, coupled to the hydrolysis of high energy phosphate bonds. So let's look at and add to this product down here. The enzyme pyruvate carboxylase. What that enzyme is looking to do is to take a molecule of pyruvate, 3-carbon molecule, and add to it carbon dioxide to make oxaloacetate, a 4-carbon molecule. So essentially, pyruvate carboxylase is carboxylating pyruvate. It's adding CO2 to it. So it's making a carbon-carbon bond. So makes a C-C bond. Okay. Now, in order to do that, what it's going to do is it's going to take ATP and it's going to make inorganic phosphate and ADP. So I need to add one product there, that PI, inorganic phosphate. So pyruvate carboxylase is going to make a carbon-carbon bond, which a carbon-carbon bond is certainly not a low energy bond. You have to put energy into that. So essentially, a ligase is going to take an ATP molecule and break it. And it's going to harness the energy that it captures from breaking that ATP molecule to make a carbon-carbon bond. So you could think about, or the way that I think about pyruvate carboxylase is it's an energy coupler. Basically, it has one unfavorable reaction and then one favorable reaction. It brings these 2 things together to make the unfavorable reaction favorable by kind of pairing these processes up. So your number 6 are your ligases, and pyruvate carboxylase is an example of this. Now, the sort of question, and I have to make a minor tweak to the exam just based on where we're completing or what material we're completing. What is most important when it comes to these 6 classes? And I'll go ahead and write on the screen here and kind of triage this information. First, ID numbers 1 through 6. So if I say, hey, the enzyme class represented by number 5 is what? I'm going to give you lyases, ligases, isomerases, transferases. The correct answer would be isomerases. Okay, so that's your first level. Second is Define number 1 through 6. So I want you to say, oh, well, number 4. Well, that is an example of an enzyme that cleaves a carbon-carbon bond. And then third would be, and this is kind of the like we're starting at a foundation and then we're building up into increasing more complex. Your third level would be where I'm asking you a question that is, um, here's a hypothetical example. So what I mean by that is I'll give you a description of what the enzyme does. And I'll say enzyme breaks ATP into ADP, P, and PI while making a CN bond. And then my question would be, what type of enzyme is this? So the way that I think about this is if you can answer these first two level questions, 1 and 2 here, then that third one should be a little bit intuitive. Now, what I want to clarify is I'm not going to say, thinking back to your slides, pyruvate carboxylase is identified as blah, where I would, and that question would only give you the name. I'm not going to do that because that to me is kind of a memorization thing. I'm instead going to ask you, and memorization is important in learning and everything of that nature, or it's always important to some degree. Um, but this question right here, this third level question, is much more impactful because then you're not associating that with pyruvate carboxylase. Instead, you're processing, oh, if I see this other enzyme where ATP is broken up, um, and another bond is made. Oh, okay, well, that's not a carbon nitrogen bond. Instead, that's a carbon sulfur bond. But the key is that ATP is broken up. Okay, well, I think that's a ligase. That to me is much more valuable. So these are the kind of three levels of questions related to the six major classes of enzymes. Now, before I proceed, does anyone have any questions about that? Okay. Well, then I am going to proceed. And I believe this is where our first top hat question of the day comes in. I'm going to go ahead and clear my writing out. Now you're going to have 45 seconds for this question. And so I'm going to read it out and everything. NAD plus and NADH are the same molecule, but one of them is the oxidized form and one of them is the reduced form. Okay. Select the true statement about these. Now, before I show the question, I want you to think about and go back in time to Gen Chem 1, where you first learned about oxidation reduction. You maybe didn't so often use the oxidized form or the reduced form. Instead, you probably talked about oxidizing agents and definitely did that in organic, an oxidizing agent or a reducing agent. And so think about for something to get oxidized, what has to take place? And for something to get reduced, what has to take place? Use that as you look at and answer this question. All right. 10 seconds left. We got 8 people. 7, 6, 4. Oh, okay. All right, so let's take a look at the correct answer. Awesome. NAD plus is the oxidized form. So NAD plus can accept electrons, and whenever it does, it gets reduced and therefore forms NADH, the reduced form. So the way that I think is helpful to think about this is look at and see, does this species have a charge, yes or no? And then kind of go from there. Okay, so now I believe, okay. So when it comes to identifying enzymes, we also have, in addition to the EC numbering scheme, we also have common names for enzymes. For example, urease. Urease catalyzes the hydrolysis of urea. Alcohol dehydrogenase catalyzes the oxidation of alcohols to their corresponding aldehydes. Now, I mentioned, one other thing that I'd like to mention is you always want to look for any sort of clue about how to classify an enzyme. This term right here, and I'm going to switch my color to, let's go with red. This term right here, dehydrogenase. Dehydrogenase is a term that is commonly associated with oxidation reduction reactions or oxidoreductases. So it's, if you're a betting person and you see a dehydrogenase, you could operate under the idea that that is an oxidoreductase. So dehydrogenase, oxidoreductase. I would kind of link those two things in my head. That will just be helpful to kind of anticipate what that enzyme is doing and what the different parties involved are doing. Now, a systematic name, a nomenclature system in which enzymes are named according to 6 major classifications of enzyme reactions, including subclasses and sub-subclasses, which is what we've gone over. And then that classification number or our EC. So this nomenclature system, the systematic name would be where you kind of identify this oxidoreductase, which is involved in the transferase phosphate group, is involved in this. You would be giving those names. You'd be giving those terms. So what that would look like would be where you see you would look at this right here, and rather than saying alcohol dehydrogenase, instead you kind of use these numbers to identify an enzyme and say, okay, one, it acts on a CHOH group, and it requires this. So you would use this kind of as a guide. You'd say an oxidoreductase that acts on an CHOH with NAD or NADP as a donor. That would be your systematic name that you would put together. Now, the EC numbering scheme makes it a little bit less wordy, and that's where you come up with that, or that's where you put together those same sorts of numbers, those identifiers for enzymes. I'm going to clear all my drawings there and go back to that slide. Okay. So the numbers represent the class. So your classification numbers are where you're giving those numbers that kind of address that phone number for every enzyme. So 1.1.1.27. Now, for most enzymes, and this is where actually I'm going to stop everything and I'm going to email this out to everyone, are cofactors and coenzymes. These are small molecules that associate with enzymes to facilitate reactions that amino acids cannot do alone. Now, does this sound familiar? So it's a small molecule or a molecule that gives an enzyme a property or a function that the amino acids alone do not. Does that sound familiar at all with respect to maybe a protein that we've talked about previously or a pair of proteins? Close, close. But let's think about our allosteric protein that we've previously talked about, hemoglobin. There was a histidine that was involved in coordinating and positioning of the oxygen, but what else was really at the heart of binding oxygen? Heme. Yes, absolutely, Milo. Heme is an example of a prosthetic group, which that's the term that we use when we're talking about a non-enzyme catalyzed reaction. When we're talking about an enzyme that catalyzes a reaction, then we want to kind of code switch and use the terms cofactor and coenzyme. So cofactors and coenzymes are molecules that give a enzyme an ability that the amino acids alone do not give it. Now, metal ions are cofactors. So metal ions like copper and iron are cofactors. Organic cofactors, so something that's non-metallic, like NAD plus or CoA. Does anyone know what CoA stands for? And it's a term that you've probably seen before, but you've maybe never seen it shortened to CoA. That CoA stands for coenzyme or coenzyme A. And you've potentially seen it as a part of the molecule known as co or acetyl CoA. Um, so an organic cofactor is referred to as a coenzyme. So I always take those two terms, cofactor and coenzyme, and I think cofactor is something that is a metal. A coenzyme is something that is organic. Now, when it comes to these two different terms and these two different molecules, they can either transiently, so kind of temporarily, or very tightly and a little bit more permanently associate with an enzyme. Co-substrates are coenzymes that only transiently associate with the enzyme. So that means they kind of like they're there for a second and then they're gone. A prosthetic group, which we've talked about prosthetic groups. Prosthetic groups are coenzymes that are more permanently associated with the enzyme. And I said that prosthetic groups are typically a little bit more associated with, um, or more commonly referred to non-enzyme or proteins with non-enzyme functions. I kind of stand by that because even here, we'll use this term that we're familiar with, prosthetic group, and then we'll say, well, prosthetic groups are coenzymes, a organic cofactor that is permanently associated with the enzyme. So I think that if you take the term prosthetic group and think about heme, okay, well, if hemoglobin had an enzyme function, well, then it would be permanently associated, or then it would be referred to as a coenzyme instead of prosthetic group. Now, I bring all this up. I'm not going to ask you questions to I'm not going to ask a question about what are the subtleties and nuances of this term. What's the difference between prosthetic group and a coenzyme? Because the difference, like the lines between them are so blurred that it's not super important. Now, with that said, I'm going to go ahead and stop right here. This is the end slide for our new material. Everything that follows this will be picked up on our next exam. Now, with that in mind, the exam that we have coming up on Monday is going to be 30 questions. About 10 of those questions are exam 2 material. Now, those 10 questions for that exam 2 material, well, I had those in the practice exam, and so some of those questions are going to be calculating the PI, calculating the charge of a molecule. What are the possible charges that a molecule can have, whether that molecule is an amino acid or a dipeptide, a tripeptide. Um, those are important things that I want you to be thinking about. Now, end my share. I think, yep, I'm going to. Done. There we go. Um, I strongly recommend going through the practice exam because it will be really helpful. It does cover kind of the same material. I've got questions that are from like the practice exam has old material questions. So calculating a PI, calculating the charge, and I walk through how to do all of those in that video demo. Um, I think that it will be very helpful. The last few questions on the practice exam might go into later on into this presentation, but I want you to think about it as anything that has to do with beyond cofactors and coenzymes is not going to be on this exam because cofactors and coenzymes is where we end. So I've got to go back and look at the actual exam and adjust some of the last few questions on that this weekend. Um, but at any rate, I hope you have a great weekend. I want you to email me if you have any questions over the weekend. I'll do my best to get back to you. I'll probably wait till later on Friday. Um, but please reach out if you have any questions. And please watch that practice video. I think that will be really helpful. Billy, there are learning objectives, and I'm sorry, the learning objectives are up on Canvas, and you know what? I posted them. I think that you see them if you go to Pages, but what I will do right now is I'll send out a message with kind of like an update on where we stopped, a link to the learning objectives, a link to yeah, I'll send that out in just a second. And the practice video is on a announcement that I posted on October 23rd. But again, I'm just about to copy and paste this basically the same message. Um, the exam, I've got to make some adjustments, and I think that the exam is going to stop on Monday at like 6 p.m. Um, there are a couple people who asked for kind of an extension of that, and I just have to confirm that that will work. But I'm looking at 6 p.m. Yes, you are allowed one note card for the exam. All right, everyone, enjoy your weekend. I'm going to stop my recording right